Now that we've talked through the step-by-step process of the sliding filament model of contraction, we need to think about what initiates contraction and what stops it once it's finished. We need to talk about how contraction is regulated. And it's regulated through free calcium ions. It's rapid changes in the concentration of calcium around the sarcomere that's going to control muscle contraction. Now, we've already talked just a little bit about calcium. Remember, we said that licorice stick protein called tropomyosin is sitting near the groove in the thin filament, and it turns out that in its resting position, tropomyosin is lying right on top of the myosin head binding sites. So even if there's plenty of ATP around and the ATP is binding to and interacting with the myosin heads, contraction cannot occur because there's something in the way of building a cross bridge, and that something is tropomyosin. When calcium levels go up in the muscle fiber, calcium will bind to the subunit of troponin called TNC. And when calcium binds, the TNC is going to change shape. That shape change is going to move the tropomyosin out of the way. Remember, troponin is bound to tropomyosin. So when troponin changes shape, when calcium binds to it, it's going to shift the tropomyosin molecule out of the way. And as long as ATP is around, the myosin heads are going to bind to the thin filament and create a cross bridge. Now on this slide, we're looking at a couple of very unusual drawings. We're looking at a thin filament and a thick filament in contact, but in a very strange point of view. So this right here represents the bundle of myosin that represents a thick filament, or at least one end of a thick filament. But we have cut through that bundle, and then we're looking at the bundle head on. We're looking at the cut surface. So each one of these brown circles is a long linear myosin molecule with heads jutting off. But we've cut all of the heads off except for one. We've left one myosin head. Now up here, the two blue circles represent the two strands of actin that make up the thin filament. But again, we've cut it in cross section, so we're looking at it end on. We're looking at the cross section, the face of the thin filament. We're also looking at the troponin and the tropomyosin. Here's the tropomyosin, the licorice stick molecule, again in cross section. And here are the three parts of the troponin: TNI, TNC, and TNT. Remember, the TNT binds to tropomyosin, the TNI binds to actin, and normally in a relaxed state, the tropomyosin is blocking this cross-hatched area right here in black. That is the myosin head binding site. So this is what it looks like in a sarcomere, from this strange orientation anyway. This is what it looks like when there is a low concentration of calcium inside the muscle fiber. Now when calcium levels are increased, this happens. We're adding calcium in to the muscle fiber. The calcium is going to bind to the TNC region and cause it to change shape. That shape change is going to pull on the tropomyosin and pull it away from the binding site. Notice it literally shifts it into the groove. It used to sit near the groove on top of the binding site, and now it's been pushed over into the groove, or pulled over into the groove might be better, and it has opened up the binding site. As long as there's ATP around, cross bridges are going to start to form and break, and contraction is going to occur. So how is calcium controlled in the muscle fiber? What is it that causes calcium to be released? What is it that takes calcium back up? Well, the answer is the nervous system. The answer is nerve impulses. Action potentials are going to do this. The release and uptake of calcium in a muscle fiber is going to be controlled by action potentials. Now, there is a specialized chemical synapse between neurons, motor neurons, and skeletal muscle. And that specialized chemical synapse is called the neuromuscular junction, or the NMJ. All a neuromuscular junction is a specialized chemical synapse. Remember, all a synapse is a point of communication between two cells, a neuron and a skeletal muscle cell. So, you want to pick up that can of soda on the table sitting next to you. Your brain sends out electrical impulses that travel down into a motor neuron, lots of motor neurons, but we'll focus on one motor neuron. That motor neuron is going to send an axon out to synapse at the neuromuscular junction with a skeletal muscle fiber. At the neuromuscular junction, acetylcholine is going to be released because acetylcholine is the neurotransmitter that is used at the neuromuscular junction. The acetylcholine is going to travel across the space in the synapse, that synaptic cleft, and it's going to bind to receptors on the postsynaptic cell, the muscle fiber. It's going to bind to the membrane, the sarcolemma. And because it's acetylcholine, it's excitatory. It's going to cause an EPSP, an excitatory postsynaptic potential in the sarcolemma. New action potentials are going to be regenerated in the muscle fiber, in the membrane. Now, the muscle membrane, that sarcolemma, this is a specialized plasma membrane. And one of the specialized features of this membrane is that it folds inwards in certain regions, creating basically passageways of membrane that we call transverse tubules or T-tubules. Everybody calls them T-tubules, so that's what we'll call them too. T-tubules actually travel around the circumference of a muscle fiber, and they occur at regular intervals down the length of that long linear muscle fiber. Take a look at the drawing on this slide that represents a single muscle fiber. The yellow represents the plasma membrane, the sarcolemma, and it has been removed from most of this cell, but we can still see it in certain regions. When we look inside the cell, we can see myofibrils, elongated groups of filaments. Here's one myofibril, here's another, here's another, here's another. What else is in this muscle cell, this muscle fiber? Well, there are multiple nuclei in here. We just can't see them. There are also multiple mitochondria. Those have been drawn in. Muscle cells perform the job of contraction, and contraction requires a lot of ATP. Cells don't store ATP, at least not in any large quantities, so it makes sense that muscle fibers would have an abundance of mitochondria in them so that they can make lots of ATP as it's needed. What else can we see in this cell? Well, we can see this network of blue. all on top of the myofibrils, running in between the myofibrils. This is sarcoplasmic reticulum. Now, this membrane, this sarcolemma, has these strange folded regions that we call T-tubules. And those T-tubules fold inwards. They fold towards the cytoplasm. And they occur at regular intervals down the length of the muscle fiber. Notice too that on either side of each T-tubule there is a region of the sarcoplasmic reticulum. This region of sarcoplasmic reticulum gets a name. It's called terminal cisternae. So there's one terminal cisterna on each side of the T-tubule. It goes terminal cisterna, T-tubule, terminal cisterna. And those three structures together make up what we call a triad. Here's the word right here. These three things are going to come in contact with each other. They're going to work together. And all these triads are going to function at the same time, essentially, to let this entire muscle cell, this entire muscle fiber, contract along its length. Now, how is that achieved? Those infoldings of the sarcolemma, those T-tubules, are going to direct the newly generated action potentials down the T-tubules. So we're going to carry the nerve impulses from the neuromuscular junction down through the T-tubules along the entire length of the muscle fiber so that we get a rapid spread of that electrical signal down the length of the fiber. The T-tubules are going to contact, remember, those terminal cisternae. And remember, those terminal cisternae are full of calcium. It's the action potential traveling down the T-tubule that triggers the release of the calcium from the terminal cisterna. Now, the sarcoplasmic reticulum has terminal cisterna in the triads. It also has the regions outside of that are called medial elements. The medial element would be this region right here. Don't worry about that term, but you should know that where the sarcoplasmic reticulum comes in contact with the T-tubule, that's called a terminal cisterna, and there's one on both sides creating the triad. The signal, the electrical signal, comes down the membrane in the T-tubule area and triggers the release of calcium from the terminal cisterna, and that calcium just floods this whole muscle fiber. It just rushes through the cytoplasm in the muscle fiber and starts this process. So let's begin with the brain sending out the impulse that you want to pick up that can of soda. The impulse travels through your peripheral nerves. We're going to focus in on one neuron that's coming into one muscle fiber. The impulse reaches that one neuron, travels down the membrane of the neuron into the axon, down into the terminal branches. We're at the neuromuscular junction. We're going to be at threshold level. Acetylcholine's going to get released from the knobs at the neuromuscular junction, at the synapse. The acetylcholine is essentially carrying that information. The information that was in the form of an electrical signal in the neuron has now been transformed or transduced into chemical information, acetylcholine. The acetylcholine travels across the synaptic cleft and binds to receptors on the postsynaptic membrane, the sarcolemma. Acetylcholine is excitatory, so it regenerates action potentials in the sarcolemma. They spread out passively, but they are directed down into those T-tubules. As they travel down the T-tubules, those action potentials, they stimulate the membrane of the terminal cisterna, and it releases calcium. Calcium floods into the cytoplasm. Calcium binds to the TNC of troponin, causes it to change shape. The entire troponin molecule changes shape and shifts the tropomyosin away from, I wrote away from the groove, but I'm really meaning away from the binding sites for the myosin heads. Now the myosin binding sites are exposed. As long as there's ATP available, contraction is going to begin. ATP, remember, binds to the myosin head. The ATP gets hydrolyzed, the myosin head cocks back. The phosphate group gets released, the myosin creates a cross bridge. The ADP gets released, the power stroke occurs. Now we're in that low energy conformation. Another ATP comes in and binds to the head, the cross bridge breaks. The ATP gets hydrolyzed, the whole thing starts again. Let's walk through it again with pictures this time. We're inside a muscle fiber, a single cell. We're looking at 1, 2, 3 myofibrils inside that cell. We're looking at just a strip of sarcolemma, of membrane wrapped around that cell. We can just see the tips of the network called the sarcoplasmic reticulum underneath that membrane. We can see an axon from a single neuron that is coming down and synapsing with the muscle fiber. That axon ends in these terminal branches, and each branch has a knob at the end that's full of vesicles that are full of acetylcholine. The action potential comes down the membrane into the branches. It stimulates the acetylcholine to be released. The acetylcholine binds to receptors in the sarcolemma. Acetylcholine is an excitatory neurotransmitter. So when it binds to the sarcolemma, it causes excitatory postsynaptic potential. It's going to regenerate action potentials, and those action potentials are going to spread out from all of those terminal branches, from all of those individual points of contact at the neuromuscular junction. And they're going to be not just allowed to spread, but they're going to be directed down into the T-tubules. That's what they're showing us here. The red arrows are diving down into those T-tubules. So we get action potential arriving all along the length of the muscle fiber all at the same time. The action potential in the T-tubule is going to stimulate the membrane of the terminal cisterna, and that's what they're showing us over here. Here's the triad. It's made out of one T-tubule and 2 terminal cisterna. As the action potential comes down the T-tubule, it stimulates calcium release from the terminal cisterna. That calcium then goes to work. It comes in, it binds to the TNC of troponin, causes the whole troponin molecule to change shape. That pulls the tropomyosin away from the binding site. As long as there's ATP around, the myosin heads are going to start forming cross bridges. The sarcomere is going to start to contract. Remember how that process happens, right? The ATP binds to the myosin head. The cross bridge breaks. The ATP gets hydrolyzed. The head cocks backwards. Phosphate group gets released, new cross bridge forms. ADP gets released, power stroke occurs. New ATP comes in and binds, cross bridge breaks. ATP gets hydrolyzed, head cocks back. Phosphate group gets released, cross bridge forms. ADP gets released, power stroke occurs. New ATP comes in and binds, cross bridge breaks. Around and around until the sarcomere is maximally contracted. Now, the last thing we need to talk about is the end of the muscle contraction. How does the muscle relax again? Well, at the end of muscle contraction, the impulse that arrived down the axon at the neuromuscular junction is going to go away. And that loss of nerve impulse means that acetylcholine will stop being released. The acetylcholine that is still in the synapse, in the cleft at the neuromuscular junction, that's going to be broken down by acetylcholinesterase. With no more acetylcholine binding to the sarcolemma, there'll be no more EPSPs generated, and that means that resting membrane potential will be restored in the sarcolemma. There'll be no more action potentials traveling down the T-tubules, so there'll be no more calcium released from the terminal cisterna. But we still have all that calcium floating around in the cytoplasm of the cell. How do we deal with that? Well, we go back to the sarcoplasmic reticulum. We go back to the blue, the network, the mesh. In the membrane of the sarcoplasmic reticulum, there are protein pumps, and those pumps are capable of pulling calcium back out of the cytoplasm and back into the sarcoplasmic reticulum. Remember, the sarcoplasmic reticulum stores calcium and releases it when it's stimulated to do so. So all of that calcium will be pumped back into the sarcoplasmic reticulum, and it will be held there until it's time for muscle contraction to occur again. Without the calcium around, the calcium is going to release from the TNC. That's going to cause troponin to change shape. That's going to release the pull on the tropomyosin. It's going to slide back over and block the myosin binding sites. That is going to end muscle contraction, and the sarcomeres will relax.